This video is a continuation of my introduction to the gross anatomy of the bones and skeleton. In part 1, I focused more on the introductory information for your bones. In this video, I'll focus on the gross anatomy of bone. The objectives of this video are to describe the gross anatomy of a long bone and a flat bone, to describe the histology or the microscopic anatomy of compact versus spongy bone, to state the functional importance of bone markings, and to compare the red and yellow bone marrow. Let's get started. Bones can be studied at 3 levels of structure. Gross anatomy, which considers the structures that can be seen with the naked eye. Microscopic anatomy, which considers those structures that can be seen with a microscope. And chemical anatomy, in which the atoms, ions, and compounds of bone and their interactions are considered. We will start with gross anatomy. There are 2 textures of bone that can be distinguished with the naked eye. Compact bone looks dense, smooth, and solid. You can see it here in this picture. Spongy bone is also called cancellous or trabecular bone. In comparison to compact bone, it looks airy and light. It is a honeycomb of flat pieces of bone called trabeculae. Spongy bone, as you can see in this image, is deep to the compact bone. In this way, the compact bone protects its fragile structures. This arrangement of internal spongy bone and external compact bone is observed in the anatomy of a typical long bone. Long bones are the most common bone in the human skeleton. These are bones that are longer than they are wide. Long bones can be divided into 3 main parts. The diaphysis, or shaft, makes up the majority of a long bone structure. The majority of this structure is compact bone, but the interior is hollowed out, forming the medullary cavity. This is where you would find red or yellow bone marrow. Adjacent to the diaphysis are the 2 bone ends called epiphyses. Here's one, and here's the other. Externally, these structures are composed of compact bone, but internally there is spongy bone. In adults, red bone marrow can be found within the spaces formed by the spongy bone. Each epiphysis is covered by articular cartilage. Remember, that's hyaline cartilage, where it provides lubrication and reduces friction in joints. The epiphysis is separated from the diaphysis by a line called the epiphyseal line, which you can see here. This is a remnant of cartilage left after interstitial bone growth during human development. That is, after we're born and we develop from young children into teens and then adults. The external surfaces of bone, that is, outside of those parts that form joints, is covered by a tough, white, double-layered membrane called the periosteum. Peri means around and osteum means bone. The outermost layer of this membrane is composed of dense, irregular connective tissue, nerve fibers, and blood vessels. This is why when you break a bone, it's often excruciatingly painful and accompanied by swelling. The inner layer is composed of bone cells that can break down or build up bone. Sharpie fibers are strong bundles of collagen, you can see them here, that connect the periosteum to bone, almost cementing it. Ligaments and tendons attach to bones through the periosteum. The endosteum is another type of connective tissue membrane, but it's internal, over all of the bone surfaces, including the trabeculae and spongy bone and the canals of compact bone. This membrane contains specialized bone cells called osteogenic cells that can change into other bone cells like osteoblasts and osteoclasts. The anatomy of flat bone is different from that of long bones. Remember, flat bones are, well, flat and slightly curved. There is no shaft or epiphysis in this kind of bone. They consist of thin plates of spongy bone, which you can see here, covered by compact bone, which is here and here. Almost like a sandwich, where the compact bone is the bread and the spongy bone is the meat and cheese inside. The compact bone surfaces are covered by periosteum, and the internal spongy bone is lined by endosteum. Although flat bones do not have a medullary cavity, red bone marrow is often found interspersed among the trabeculae of the spongy bone. Any parts of a flat bone that form a joint will be covered with articular cartilage. Again, this is hyaline. Now, in your cranial bones, your spongy bone has a special name. It's called diploe. Diploe. And that is spelled Diploe umlaut. Red bone marrow, also called myeloid tissue, that's myeloid, myeloid tissue, has stem cells for the production of blood cells, so it is involved in blood cell production. Remember, there's a special name for that, hematopoiesis. Before the age of 7, all bone marrow is red bone marrow because of the increased need for red blood cell production. After the age of 7, red marrow is slowly converted to yellow marrow in most places. In adults, red bone marrow is found only in the vertebrae, hips, breastbone, ribs, skull, and in the heads of the femur and humerus. A bone marrow transplant is a medical procedure that is performed when a person's red bone marrow has been destroyed or damaged by disease, infections, or chemotherapy. The procedure involves replacing damaged or destroyed red bone marrow with red bone marrow from a healthy individual. I have included a link of a video showing a bone marrow aspiration from a healthy individual. When you have time, take a look at this video to see what is involved. Yellow marrow is formed after the age of 7, and by the time we are adults, most of the bone marrow in the medullary cavities of our long bones is yellow marrow. This marrow consists of blood vessels, adipocytes, and mesenchyme. In instances of severe blood loss or fever, this marrow can be converted to a red marrow, however. Adults have on average about 5.7 pounds of bone marrow, with about half of it being red bone marrow. So let's see what you remember. The terms long, short, flat, and irregular are used to classify bones based on their, would that be tissue content, function, shape, or weight? I know we covered this in our last video, but hopefully you remember the answer. Shape. Long, short, flat, and irregular are terms used to classify bones based on their shape. The most important characteristic for designation of a bone as a long bone is its elongated shape, its total length, its length relative to other bones, or its location in the body. Its elongated shape. That's right. Which of the following is not a function of bones? Would that be protection, support, mineral storage, or transportation? Transportation. That's right. Transportation is a function of blood. The principal component of bone that contributes to its hardness is hydroxyapatite, collagen, osteoid, or organic. Hydroxyapatite. Good job. If you look closely at a bone, you'll probably notice that although it looks smooth from afar, in actuality it has a number of projections, bulges, depressions, and openings. These features are called bone markings. Bone markings are critical to the identification of individual bones and help us understand the functional aspects of human anatomy. Familiarity with bone markings is an essential tool for doctors, surgeons, orthopedists, physical therapists, radiologists, and forensic scientists, among others. Your bone markings can be grouped into three categories. Projections and bulges are places where ligaments and tendons bind the bone, and the constant pulling by the ligaments and tendons results in a bump. Projections and bulges may also allow a bone to form a joint. Joints are also called articulations, by the way. Depressions are places where the bone surface curves in. This forms a basin. These are typically areas where blood vessels pass along the surface of a bone. Openings are holes in a bone. They are places where nerves and blood vessels pass through the bone itself. Now let's look at some common bone markings and examples. An understanding of these characteristics of bone will help you do well on your lab practical and exams. This information may show up as a multiple choice question on exam 2 and on your final. The first bone marking is called a tuberosity. This is a moderate prominence in which muscles and connective tissues attach. An example of a tuberosity is seen here. This is the deltoid tuberosity, where the deltoid muscle actually attaches to the humerus. Next, we have a crest. Crests are raised or prominent parts of the edge of a bone. Connective tissues attach muscle to bone here. A great example of a crest is this one on the pelvic bone. This is called the iliac crest. Next, we have trochanters. Trochanters are large prominences on the side of a bone. Some of the largest muscle groups in the body and dense connective tissues attach here. Examples include the greater and lesser trochanter of the femur. Finally, a line is a narrow ridge of bone, but it's much less prominent than a crest. Examples can be found on the femur. There's a ridge of bone, a line, that goes from the greater trochanter to the lesser trochanter. It is called the intertrochanteric line. There's another line on the femur called the linea aspera, but it is not pictured here. Tubercles are small rounded prominences where connective tissues attach. Examples would include the greater and the lesser tubercles of the humerus, which are depicted here. Epicondyles are small prominences, that is, bumps, that sit on the top of a condyle. They are attachment points for muscle and connective tissue to bone. Examples include the medial and lateral epicondyle of the humerus. Spines are sharp, slender projections. They are usually very pronounced and functionally serve as attachment points for muscles and ligaments. A great example of a spine would be this one. This is the spinous process of a thoracic vertebra. Generally speaking, anytime a portion of a bone or a part of a bone sticks out from the bone surface, it will be called a process. On the last slide, we looked at projections that are sites for muscle or connective tissue attachment. Now let's look at projections that form joints, aka articulations, articulations, articulations. First, we have a head. The head is a rounded expansion of a bone that forms part of a joint. Usually it sits on a narrowed region called a neck. Great examples of this would be the head of the femur, which you can see here. So here's the expansion, and then this is the neck. And the head of the humerus, which you can see here. From this view, it's a little harder to see the neck. Next, we have facets. Now, when I think of facets, I think of gemstones. A facet is kind of like that smooth surface that reflects light on a gemstone. On a bone, a facet is the same thing. It's a flat, smooth surface. And this flat, smooth surface forms a joint with another flat bone or smooth surface. So a great place to find these are on your vertebrae. So this is our thoracic vertebra. And I hope you can see that there's a flat surface here, and there's a flat surface here. There's also a flat surface here and over here. These are all facets, and they interlock with adjacent vertebra so that the bones can move with respect to another. It gives flexibility along the spine. In this image, you can see how the facets fit together for these lumbar vertebrae. So you can see that you have an articular process, a superior articular process that interlocks with the inferior articular process of an adjacent vertebra. Condyles, sometimes pronounced condyles, are large rounded prominences that bear the brunt of force exerted by a joint. Examples can be found on the femur, the tibia, and here on the skull. These bumps that look a little bit like half moons here, these are the occipital condyles. They form a joint with the first cervical vertebra. Our last projection is a ramus. A ramus, which is depicted here, is simply a curved arm-like bar of bone, and it gives structural support to the rest of the bone. The last two types of bone markings are the depressions and holes. Let's take a look at some common ones. First, we have grooves. Grooves are furrows in a bone surface that run along the length of a blood vessel or nerve to provide space to avoid compression of that structure, the blood vessel or nerve, by adjacent muscles or external forces. An example of this would be the radial groove, which hopefully you can see here. It's the thing that looks kind of whitish on this rather dirty model. But the radial nerve basically passes along the humerus at that point, and this space helps prevent us from crushing that nerve. Fissures are open slits in bone that usually house nerves and blood vessels as well. So an example would be the superior orbital fissure, which you can see right here. Foramina, the singular is foramen, are holes through which blood vessels and nerves pass. Examples would be the foramen magnum, that rather large hole on the on the inferior aspect of the skull. And then you have these smaller foramina, which are called the mental foramina. So here's one, here's another. A notch is a depression in a bone that may stabilize an adjacent joint in a bone. The articulating bone may slide into and out of the notch as part of the movement of the joint. So a couple of examples can be seen here on the head of the ulna. This is the radial notch. The head of the radius, which you can see here, fits into that notch, forming the radial ulnar joint. You also have the trochlear notch here. The trochlea of the humerus fits here, forming a joint between the humerus and the ulna. A meatus, depicted here, is a tube-like channel that extends within a bone and may provide passage and protection to nerves, blood vessels, and sound. This meatus is the external acoustic, sometimes called auditory, meatus. Sinuses, which are not depicted on this slide, are air-filled cavities within a bone. They help sound resonate in the skull, and they also make the skull a bit lighter. And finally, fossa are shallow depressions in a bone surface. They may receive an articulating bone or support brain structures. An example would be the olecranon fossa, which the olecranon process of the ulna fits into, right here on the distal end of this humerus. Let's try a couple of review questions. Depressions on bone include, would that be fossae, trochanters, tubercles, or tuberosities? Fossae. An example of a projection on a bone, that is the site of a muscle or ligament attachment, is a, would that be meatus, head, condyle, or trochanter? Trochanter. Good job. The last thing that I want to consider in this video is the part of bone anatomy that we cannot see with our naked eyes. If we were to use a microscope to look at the microscopic anatomy of bone, it would look like a little maze of concentric circles, kind of like what you see down here. The basic structural unit of a compact bone is the osteon, aka the Haversian system, and you can see in this picture that several of them have been highlighted yellow. This structure is composed of weight-bearing column-like tubes called lamellae. Over here you can see what they kind of look like if you pull them out. The collagen bundles, represented in gray on this slide, run in opposite directions. This gives bone some resistance to twisting forces. In the middle of each osteon is a large hole or opening, which you can see here and here. This is called the central or Haversian canal. This structure allows blood vessels and nervous tissue to penetrate the interior of a bone. The central canals of compact bone are connected to each other and to the blood vessels in the periosteum via these perforating or Volkmann canals, which run at right angles to the central canals. In between the lamellae are small cavities called lacunae. These house special cells called osteocytes. Osteocytes help maintain the bone matrix and communicate with each other and other bone cells called osteoblasts and osteoclasts through hair-like canals, which are depicted here, called canaliculi. The trabeculae of spongy bone, depicted here, do not contain osteons. Instead, they have irregularly arranged lamellae with osteocytes and canaliculi, which you can see here. Blood flow, and thus nutrients, come from capillaries, tiny hair-like blood vessels located in the end ostium. Just a note about trabeculae in spongy bone. Typically, trabeculae align along the stress lines that are put onto a bone. So wherever the stresses are coming from, that's how they will arrange themselves. So if you imagine that a pelvic bone is right here, that means that there is stress being put on the head of the femur. Primary medial trabeculae align themselves in this direction to try to resist that stress that's being put on the femur by the hip bone. Primary lateral trabeculae align themselves in this direction to resist compression and compressive forces. At this point, you've heard me mention the names of several bone cells at different points during this lecture. Let's take a look at what they are and what they do. There are 5 major cell types in bone. You have the osteogenic cells, osteoblasts, osteocytes, bone lining cells, and osteoclasts. Osteogenic cells are also called osteoprogenitor cells. These are stem cells located in the periosteum and endosteum, and they change as needed to become osteoblasts or bone lining cells. Osteoblasts are bone-forming cells. They are the bone builders. These cells are mitotic, which means they reproduce, and their main job is to secrete osteoid. 90% of the bone protein that they are secreting is collagen and calcium binding proteins. In addition to secreting these proteins, they're secreting proteoglycans as well and other substances necessary for making the bone matrix. When osteoblasts become surrounded by enough bone matrix, they mature to become osteocytes. So these are osteocytes are mature bone cells. They're located in the lacunae, and their job is not to secrete lots and lots of bone matrix, but rather to maintain the bone matrix. They communicate mechanical stimuli to osteoblasts to stimulate bone building and to osteoclasts, which break down bone. The process of making bone and breaking down bone is called bone remodeling. Bone lining cells are flat cells that line bone surfaces. They are believed to help maintain the bone matrix. When they are on the external surface of a bone, they are called periosteal cells. When they are on the internal surfaces of a bone, such as what you see in this diagram, then they are called endosteal cells. The last type of cell is called an osteoclast. These are bone destroyers. They break down or resorb bone. Resorption is the funny term that we use for the breakdown of bone. These cells secrete acids from this ruffled border here, and those acids break down hydroxyapatite crystals. They also secrete enzymes from that ruffled border that break down collagen and proteins. The cells then reabsorb the digested bone products and recycle them. Let's try a few questions just to wrap this video up. A group of concentric rings of bone matrix comprising the functional unit of compact bone is called, would that be a lamella, an osteon, a pillar system, or Sharpie system? An osteon. That's right. It's the group of concentric rings that's important here. One ring would be a lamella, but the group is an osteon. Adjacent osteocytes communicate via gap junctions found within, would that be lacunae, Folkman's canals, Haversian canals, or canaliculi? Canaliculi. So remember, canaliculi are these thin hair-like connections between osteocytes that allow communication between the osteocytes within bone, as well as communication with the osteoblasts and osteoclasts that are on the external surfaces of bone. You did it! You made it to the end of the video. In this video, I described the gross anatomy of long bones versus flat bones. Remember, your long bones have a diaphysis, 2 epiphyses, medullary cavities, articular cartilages. They're surrounded by periosteum externally and endosteum internally. Remember, your flat bones don't have any of that. It's little more than a bit of spongy bone that is sandwiched between 2 layers of compact bone. The compact bone is covered by periosteum, and then internally you have endosteum as well. We also talked about the microscopic anatomy or histology of your compact and spongy bone. We talked about structural differences between these two types of bone. Remember, compact bone has Haversian systems with lamellae, canaliculi, osteocytes, and central canals. You also had the Folkman canals in play. Spongy bone doesn't really have that. Remember, spongy bone doesn't have osteons. There's no central canal. Instead, you have irregular lamellae, you have the canaliculi and the osteocytes, and blood and nutrients comes through the bone via small, tiny capillaries that are in the endosteum. I also talked about the functional importance of various bone markings. We talked about foramina, we talked about condyles, we talked about spinous processes, and all sorts of bone markings. Remember, for your lecture exam, this material is usually a multiple choice type question where I'll ask you to tell me what the purpose of the different types of bone markings are. In your lab practical, you'll have to actually identify some of them. Finally, I talked about the differences between red bone marrow and yellow bone marrow. Remember that red bone marrow is mostly associated with the production of blood cells. Yellow bone marrow mostly stores nutrients in the form of fat. So, I hope this video was helpful to you. If anything was unclear or you have any questions, please don't hesitate to reach out to me. I am more than happy to answer your questions. In the next video, we'll talk about how bones come about from the time that we're in our womb to the time that we actually become adults. Did you know that many of your long bones, for example, actually start out as cartilage models when you're in the womb? And that many of your flat bones actually start out as membranes? I'll get to that and a lot more in the next video. See you there.